Hello and welcome back here live from the Bella Center in Copenhagen, Denmark. We are about to start a very historic moment in the history of the transplantation community, in the history of the global transplantation community, because in about 5 minutes we are going to connect this physical congress here in Copenhagen live with the Stanford University School of Medicine. And if you've been watching our live broadcast, either on YouTube or Facebook before, this is the outfit, this is my Copenhagen suit. But earlier this year, I had the honor to visit Dr. Waldo Concepcion, one of the chairs in Stanford. And when I met up with him, he told me I couldn't leave without my Stanford merchandise. So in his honor, I will now transform myself into a proper Stanford School of Medicine student and be part of this session as well. So hold on for a moment. Jacket. Let me pick up my Stanford sweater. Stanford School of Medicine, I should say. It was a gift. There we go. And then obviously we finish that off with a nice Stanford hat. So, this is my transformation, and I hope it's symbolic for the transformation that's going to happen right now. Because just imagine what could happen if we, in the next 2 hours, combine the most inspiring minds in transplantation together with the incredible driving force for change in Silicon Valley. And that's, if you're not familiar with Stanford University, it's in California, in the heart of Silicon Valley. It's surrounded by all these great tech startups, and even more and more of these startups are going into the field of medicine, biotech startups. And what we will do for the next 2 hours is actually follow that journey from an idea, from a research insight, maybe from a patent, and see how you can preserve that, and then turn that into a prosperous business, and through that way, not only save lives or procure more organs or get people off the waiting list, but also have an even bigger impact on society, because your research insight, your academic knowledge is not only in your papers, not only in the academic community, but gets transferred into value for everyone involved and gets a chance to scale up throughout the world. So what can you expect in the next 2 hours? We're going to start in any moment now in a session here in Copenhagen chaired by Stefan Schneeberger, who is since 2 days now the past president of ESOT. And he will kick off and introduce the session right here. And then we'll connect to Stanford Live, where Randall Morris and Waldo Concepcion will chair the session from there. And what is a very big honor for this meeting is that we will have an official welcome by the Dean of the Stanford School of Medicine, Dr. Lloyd B. Minor. He's actually right now, it's early morning in California, he's joining the meeting over there, and he will officially kick off the meeting and open that up. And then we'll go through a series of speakers, short, inspiring talks. We'll introduce each speaker one by one, and then they have 10 minutes to give you some key takeaways of what they have learned on their journey from an academic idea or insight into true value or a business startup. After 3-4 speakers in Stanford, we'll have 2 speakers right here with us in Copenhagen. And they're both from biotech startups that have been remarkably successful. You might have seen Dr. Edward Chong before here in the studio, chatting with Stefan Schneeberger on Monday morning. And also John Piano, who is the founder, CEO, and chairman of Transplant Connect is here, a platform that is a software device for transplant coordinators and making the transplant coordination much more simple. Then we turn back into Stanford and hear more war stories of entrepreneurs in the biotech startup sector, and actually then gather all these ideas into key takeaways. It looks like the session is starting. Let's get into room A3.
Don't have a picture at all, or you're looking at a slide? What are you looking at?
We can see you.
You can see me?
Yes.
Very good. Okay, we're getting ready. Maybe a few more minutes. You let us know when we can get started from your end.
Okay.
So all good? Yeah.
All right, so I guess we were just looking at the last technical rehearsal. It's officially 6:30 right now. I'm having my eye on room A3 over there. It's in the other side of the hallway, and I noticed Stefan on stage. I can't hear what he says, so I said we're starting. But if you're just joining us on Facebook, welcome. This is a truly interactive session, so you can also send in your questions via YouTube and Facebook, and I will relay those questions on your behalf, because at the last 20 minutes of the session, we'll have an interactive Q&A with both the audience here in Copenhagen and the audience in Stanford. So I hope that we will see the situation that the audience in Stanford asks questions to speakers here in Copenhagen, the audience in Copenhagen asks questions to speakers in Stanford, and you as an online audience send in your questions via Facebook and YouTube so we can relay them as well. I'll go inside the room, and if I see your comments, I'll raise my hand and share your question on your behalf. So that's very exciting stuff, and this is part of a much bigger project where we are trying to connect the global transplantation community throughout the world via ESOT's educational platform, Transplant Live. And if you're not yet a member of that, you should sign up right away. The URL is esottransplantlive.org. If you sign up for the first time, it's just a small yearly fee, and it gives you access to, first of all, this full 4-day, 24-hour live stream that's going on in the conference right here, and an extensive library of all the presentations that have been recorded in the last 3 congresses, all easy navigatable, easy browsable. And most importantly, you can earn online Cme credits, so your permanent education credits can be obtained by participating in the online education platform in specific courses organized either by organ, or there's an ethics course, or there's a whole broad range of courses that you can participate in and make sure you're up to par in the most latest insights. So we are now getting ready to take you into room A3. If you hear some rumbling and noise in the background, there's actually a cocktail bar in the back of that room, because by now it's 6:30 here in Copenhagen, so it's also the YPT reception taking place in the back as well. So the young transplantation professionals are joining this session and be on the forefront of how this transplantation community is connecting throughout the world. Now, if you're curious to learn more and you haven't had a chance to hop over to ESOT Transplant Live, then make sure you'll be back with us tomorrow morning. Early in the morning, we kick off day 4 of the ESOT Congress here in the little studio, and I will have a bike ride with Thomas Rash, who was a member of the Scientific Program Committee for the Congress right here, and is also a board member of the YPT. There they are again, the young professionals in transplantation. And I will have a chat with him about some of the sessions they organized here in the Congress, what's hot and what's happening for the young professionals. And I will also have a chat with him about what's hot and what's happening here at Congress, being here for 3 days. Finally, we round off tomorrow morning with the presidential session. Vasilios Papalois, who is the recently elected president, will join me here in the studio and look forward to his dreams and ambitions for the next 2 years. And then we will take you straight into the last plenary, the presidential session, as it were called. I hear we go straight to stage in about 30 seconds, so let's get into room A3. Let's connect live to the Stanford School of Medicine. I'm ready for it, and if you are anywhere in the world, let us know where you're watching from right now here on Facebook. I see Shagib Arafin is doing that. Great. Thanks for participating. Keep that up. If you have a question during the session, keep posting it, and I will relay them on your behalf. Have a great session. I would say, Stefan, take it away.
Hello, everybody. Hello, delegates in the room. Hello, delegates over the Atlantic in Stanford. Welcome you all to a unique and new event on many levels for us here in Copenhagen. I'm on the stage here together with Edward Chong, the CMO of Vitaris, who will be one of the speakers telling us his story, and with John Piano, who's the Chief Executive Officer, but also the founder of Transplant Connect. Thanks, first of all, to Randall Morris, Walter Concepcion, and Annalisa Ponkia for putting this experiment together. Before I go on and introduce the idea of the session, I'd like to pass on the word to Randall Morris. I think he's standing at the microphone over there in Stanford. Hello, Randy.
Hello, Stefan. Good to see you. Everything seems to be working very well.
Wonderful. You have a good gathering, Randall?
Standing remotely.
Very good. Well, thanks again for doing this. You were instrumental in putting this together. Let me briefly introduce the idea that came across when we started putting this together. I think if we take a step back and look at the history of transplantation, we all do acknowledge that great achievements were made in the 60s, 70s, 80s, and 90s when transplant became a clinical routine. And then in the early 2000, the success rate actually went up to a level that was quite satisfactory, but it was more related to short-term success. What was left unaddressed were the long-term outcomes after solid organ transplantation, and they're much less satisfactory. So that resulted in a difficult situation with the lack of endpoints for clinical trials to develop new technologies and also new drugs to further advance the outcomes in transplantation, while people were suffering and while we continue to lose organs. Just the late endpoints are not valuable, they're not fitting for clinical trials, and that's the situation I think that we're stuck in. So what we see now is that on one hand, the field of transplantation is maturing, and the large industry, at least, is losing somewhat its appetite to invest further in the field. Whereas there are important problems to be addressed, and I think that leaves a gap, that leaves an opportunity, to formulate it more optimistically, for new and possibly also smaller industry to come in and more specifically address the remaining important issues in transplantation. So it's interesting that we're now moving from a relatively mature field with a strong industry interest in investment to a field that comes back, so to say, to its roots, where there's more a startup feeling, a startup atmosphere, and a lot of opportunities. And this was sort of the original idea that we picked up, and we said, well, if this field is moving to sort of a startup phase again, then we have to address and embrace the circumstances. And there's maybe no better region on the globe to involve at that point rather than Stanford University, and its proximity to Silicon Valley and its embracement of startup. Since the startup idea, I guess, is implemented in this region. And that is when Randy, Waldo, Annalisa, and I started putting this together. And the idea is to deliver to you here and back also to Stanford the idea of the history, the concept of startup companies. And we want to do that by delivering stories of people who went through that, and they can explain best what it would mean. And hopefully that is inspiring to many of you to be brave enough and eventually make a decision to say, yes, I'm going to pick up the idea. There's something, there's a product, there's an idea that I would like to bring to the market. It's a long way. I'm sure it's a very difficult way, but it's worth doing. And that is the subject that we want to address today. So that will be it from my end to start with. Randy, I'll pass it on to you to continue and introduce the session from your end. I see it's Waldo now coming to the microphone. Hi, Waldo. Welcome.
Good morning, Stefan. How are you doing?
We're all well. We're all well. Happy to connect with you.
Well, we're so excited. And the most important is when we're here in Stanford, our goals and our visions are always to heal humanity through research and innovation. And this is something very dear to us. That is not just about the fact of doing practice in medicine, it's how can we make it better in every single moment. And I'm so glad that we started this relationship because we want to foster this across countries, continents. We're all in the same goal to make humanity better. And in Stanford, we have been blessed for the last years of having tremendous impetus on bringing innovation, bringing most important focus on what we want to change the world of medicine, and that's to precision health. And I'm so happy and so proud to have the honor to introduce our Dean Lloyd Minor, that is the Carl and Elizabeth Newman Dean of School of Medicine. And this is very important for us that with his leadership in Stanford Medicine, we have established a strategic vision. It's not just about doing it. What is the vision for the future to lead environmental revolution in precision health? If we think about it, it's a fundamental shift that is more proactive and personalized healthcare that empowers people to lead healthy lives. We're not in the business just of treating disease. We're trying to make patients healthy. And that's a concept that precision health comes about. Dr. Minus also has a degree as a professor of otolaryngology, head and neck surgery, and professor of bioengineering and neurobiology by courtesy at Stanford University. He has more than 140 publications and chapters, and Dr. Minor is an expert in balancing the inner ear disorders. And the aspect that I'm proud that all the country recognized that he was elected in 2012 at the National Academy of Medicine. It's a great honor for us to have him as our Dean, Dean Minor.
Well, thank you very much, Waldo, for that really kind introduction. And Waldo, I want to congratulate you and also Randy Morris and Stefan Schneeberger from ESOT and Copenhagen for organizing this amazing conference. I think it's a credit to technology that we can be connected so well. And I must say, I breathed a sigh of relief this morning when everything was working. But these connections are getting better with time. You know, I think the focus of your conference today is very timely, and that is how do we get discoveries into the marketplace? How do we get discoveries from the laboratory and into companies and ultimately broadly to people? I think that's the key message, is that companies are never going to do away with the necessity to have outstanding research done at places like Stanford, and certainly they're not going to in any way supplant the outstanding care provided by Waldo, by you, by Mark, by all of our transplant surgeons here at Stanford. But what they can do is accelerate the process of getting from an early discovery to actually getting to a therapeutic or a preventative strategy or a diagnostic that can be used to help in patient care. And talking about how to facilitate that process, I think is very timely for a conference like this. I also want to say how excited I am about the work that is going on here and internationally in organ transplantation. And of course, Waldo, because of you, because of Carlos, Mark, so many other people that are a part of the transplantation team here at Stanford, you've really set the bar very high for the surgical outcomes in organ transplantation. And in so doing, you focused a lot of attention on the need for a better understanding of basic immunology, of organ transplant rejection, and a lot of the advances in those areas have come about because of the great work that you and your colleagues have done. I made remarks a few days ago at a symposium where I tried to make the connection between what we do clinically and what we do in research labs. You know, Stanford as a university is about research and teaching. Of course, we're a research university. But our academic medical center, very importantly, has the goal of delivering outstanding patient care. That's one of the three pillars of our academic medical center, and indeed every academic medical center. How then can we look at, over time here at Stanford, how that patient care mission has helped to support and drive advances in research and teaching? And one of the examples that I mentioned was going back to the work of Dr. Shumway and the first heart transplantation, then following that with Bruce Wright's, the first combined heart-lung transplant. And what Dr. Shumway and his colleagues discovered very early on, 50 years ago, was the surgical techniques were challenging. And being a surgeon myself, although not operating anymore, I'm not in any way trivializing the difficulties and the importance of what we do as surgeons. But what proved to be the biggest challenge ultimately for the outcomes in cardiac transplantation was rejection. So Dr. Shumway and his colleagues, because they were brilliant surgeons, very early on mastered the techniques of transplanting a heart. But what was limiting the success and the survival in their patients was not the surgical technique, it was the rejection of the organ. And that drove an entire focus here at Stanford and elsewhere, but I think first here at Stanford on building out human immunology and research in human immunology, focused on understanding the immune system and then on applying that knowledge to understanding the phenomenon of rejection of transplants. And today, I think arguably, we've got the best immunology training programs and research programs in the country. But I think in no small measure, the stimulus for building those areas of focus here at Stanford came from the early work in cardiac transplantation, then of course closely following that, other solid organ transplants as well. And finally, I just want to say, in thinking about this process of commercialization, I would come back to what I said before, and that is commercializing things is extraordinarily important. There's never been a better opportunity than there is today to do that. But commercialization would be meaningless and not successful without the type of discovery-led research, without the type of outstanding patient care that goes on here in Copenhagen and in so many other places every day. So we want to accelerate the translation of that research into products through commercialization. We also want to affirm the need to continue to support the discovery-driven research and the amazing clinical care, Waldo, that you and your colleagues provide every day. To our colleagues in Copenhagen, it's a great pleasure to meet you and see you remotely. I just want to say we at Stanford have many wonderful interactions in Denmark. We do a lot of work with a Danish biobank and Mads Melby, who directs the Danish biobank, which I think our colleagues in Denmark are well aware is one of the premier biorepositories in the world in terms of the treasure trove of information. And we've had the privilege of collaborating with Mads and his colleagues on the informatics and on leveraging and discerning the information embedded within that tremendous data set. And we look forward to continuing our collaborations. Thank you very much. I wish you a very successful conference today, and congratulations. Thank you.
If I may, Waldo, very briefly, I'd like to just thank Dean Miner for this introduction, for supporting the idea, embracing the concept of what we're doing today. So thank you very much, and thank you for getting in contact with us. Stanford University is representing the spirit of innovation so well that we're very grateful to be connected and grateful for your support. Thank you.
Proceeding with our program, it's an honor to also present today one of the epitomes and example of what Stanford has done over the years. When we talk about Dr. Randall Morris, we talk about Stanford and we talk about a giant in the field of immunology and transplantation. He has been at Stanford for 30 years as an undergraduate, medical student, surgical intern, founder and director of the Laboratory of Transplant Immunology in the Department of Cardiothoracic Surgery, and away from Stanford at the University of Wisconsin for his surgical residency and postdoc, and in Basel, Switzerland, as a global head for the transplant drug discovery at Novartis. Along the way, he also led the teams and collaboration and discovery of, I just wanted to make a pause and say, every day that we take care of transplant patients, we had to think about Dr. Randall Morris. He was capital in developing mycophenolate mofetil, sirolimus, tofacitinib, someday I'll learn how to say it, that helps give us tools for preventing transplant rejection. He and Claire Gregory also co-invented the first effective drug-eluting coronary artery stent. Dr. Morris will be speaking about university and startups, engines of change. Dr. Morris, good time.
Thank you very much, Waldo, and welcome to all of you in Copenhagen. I'd like to thank the ESOT for embarking on this very creative session, and I'd like to thank Waldo Cheung very much for helping lead this to fruition. Waldo is a professor of surgery and pediatrics at Stanford. His interest is in pediatric transplantation, immunomodulation, stem cells, liver heart transplantation. And it seems like just yesterday, Waldo, that you got here. I remember when you joined our group. My task today in 10 minutes is to talk about universities as the engine of change for startups. And I guess I'll start with a comment that I heard somebody said, which was that in order to get tenure at Stanford, you have to found a startup. Well, I'm the first speaker. I did get tenure, but I have not found a startup. I'll tell you a little bit more about me because ESOT wanted all the speakers to talk about their stories, but I really want to talk about you there in Copenhagen, you who are thinking about a startup, and I want to try to encourage you to think of key questions you should ask yourself before considering doing a startup. For me, Stanford was a very good preparation for possibly starting a startup. In those days, there was a very great dichotomy between basic science at Stanford and pure clinical patient research. In the early 2000s, mid-2000s, Stanford, that changed dramatically. Translational medicine was brought in, and Stanford actually has infrastructure for students, fellows, faculties who want to know about and who want to implement a startup. You can see these infrastructures online. One is called Spark, S-P-A-R-K. The other is called CERSI, C-E-R-S-I, which is regulatory work. The other new one is Alliance for Innovative Medicine, where Stanford is collaborating with pharma, and Stanford does have a relationship with UCSF in San Francisco with the Rosenman Institute. So there are many entities which are there to help people get started if they want to do a startup. When we were doing our research on drug discovery, drug development at Stanford, we were rather unusual because we were doing applied research. But that really set the stage for what we did subsequently and my interest in startups and my thinking about it. We wanted to develop new drugs, as Dr. Miner pointed out, as essential agents for transplant patients. And I was recruited then to Novartis in Basel, which was a total change culture, sort of like drinking water from a fire hose. It was really a dramatic education. I had the advantage of starting out in discovery, which is the early part of drug development, moving then to translational medicine, all for transplantation, and then moving to development, and then finally moving to business development, pricing and reimbursement. So I got a postdoc in pharma business. Now, before I end talking about me and talk about you and the audience in Copenhagen and things you should consider about a startup, let me say that we, meaning I and another group, have considered doing a startup, but we're very well aware of the fact that the statistics for biotech generally are that 92% are not profitable, and only 2% give the investors a good return on their investment. So doing a startup is hard, difficult, and very risky. Well, if you're considering doing a startup, you should really consider a question that I ask venture capitalists who asked for my opinion on potential startups. And the question I asked the venture capitalists is, did you ask the people who are pitching their idea to you what the claims on their label for their drug would be? I'm going to focus on therapeutics. Other speakers will talk about diagnostics. 95% of the potential founders don't consider the claims on the label. The claims are the unmet needs, the indications, the phase 3 primary endpoints. So you need to think about that before you do your therapeutic startup. You need to think about the mechanism of the disease. Do you really know the pathways that lead to disease at the cellular molecular level? Does the target for the therapeutic block a pathway which is non-redundant? In other words, does it block a key pathway leading to that disease? So these are all the questions you should be asking yourself before considering moving ahead. The drug or therapeutic you want to develop can be a first-in-class novel drug for an unvalidated target, which can be exciting, but it's very risky because you don't know whether it'll be effective and you don't know the toxicity. You can decide to take the route of a validated target, making a second or third-in-class drug where you know the target really does prevent or treat the disease. But you have to remember that your therapeutic has to be best in that class. And the key message is that your therapeutic has to be better than the standard of care, not today, but 5 or 10 years from now when you launch it, which requires quite a bit of guesswork. Let me talk about one of the problems that biotechs and pharmas have in being more effective. And this gets back to universities. Unfortunately, studies have been done recently. Amgen and Bayer have reviewed hundreds of publications of biology, and only 11 to 22% of the data were repeatable. So you have to ask yourself, are your lab data real? Are they rock solid? I'll quote from a book in 2019 called Fraud in the Lab. Quote, 75 to 80% of publications are not publishing data that is repeatable. And pharma relies on university knowledge, basic science to do their work, so they need the data from university to be repeatable. Another interesting little sidebar is the retraction index of publications. Maybe you would like to guess which journals have the greatest retraction index. I'll tell you. Number one is New England Journal, most retractions. Number two is Science. Number three is Cell. Number four is Nature.
Oh.
So don't be too disappointed if you don't get your publications in those journals. Let me just very briefly talk about a key area you also need to consider when you're doing a startup, and that is the exclusivity of the therapeutic. It should have good intellectual property. It should be patentable. Patent should be broad, non-infringing, and basically a patent needs to be novel, useful, non-obvious. That's a very quick summary. I think, in my opinion, if you're doing a startup biotech therapeutic, and you only have one or two assets, not only is it good to have your patent written, probably by your university, but I would suggest you get an outside patent litigator to review the claims on your patent, because the litigators are the ones who are going to have to defend your patent. And they're the ones who are going to see any weaknesses in the claims of your patent. I think my time is getting up now, so I will just conclude with a research organization's top 10 reasons why startups fail. These are not necessarily biomedical, they're all startups. And I hate to be negative for you there in Copenhagen, but I don't want you to embark on a very hard journey unless you have really done your homework before you do your startup. So the number one reason that startups fail is no market need, that's 42%, ran out of money, 29%, not the right team, 23%, outcompeted 19%, price cost issues, 18%, product without a business model, 17%, ignoring the customers who are the doctors, consumers who are the patients, and the competition, 14%, loss of focus, 13%, and disharmony among your team members, 13%. In conclusion, most drugs fail, most startups fail. You and your team need to quote Paul Ehrlich, the father of immunology and chemotherapy back from the century. You need skill, patience, money, and most of all, luck. Thank you. And next, I want to introduce Ms. Deepa Rich, who is a partner at Goodwin legal firm, which is a biotech legal firm specializing in startups all the way through to initial public offerings. It's my pleasure to have Deepa tell us about the life science entrepreneurship stories of success and failure. Welcome, Deepa.
Thank you. Thank you very much. Well, it's a tough act to follow. Well, thank you all for having us. First, thank you to Stanford School of Medicine, and thank you, Dr. Morris, for that very kind introduction, and also to the European Society of Transplantation, Organ Transplantation. So my name is Deepa Rich. I'm a partner at Goodwin Procter here in Redwood City. I'm a corporate biotech lawyer. I do not have a science background, but I have dedicated my practice of law to working with life science companies. I'm a life cycle lawyer, meaning I represent companies from when it's just an idea, and we don't even have a company yet, through to when a company has an exit, potentially an M&A or an IPO, and then represent the public company as well. So through my years, I've worked with a lot of companies and have an idea of what is successful and what has not been successful over the years. To prepare for this, I actually reflected on, I think we've formed over hundreds of life science companies. So to try to think about who was successful, talk to a handful of our founders, and try to see what the common themes were. I came in here this morning and I kind of shared some of that idea, to kind of just see how it floats. And I won't say who it was. Another one of our speakers said, well, it's all great, but I really think it's all luck and timing. And I thought, well, there goes my entire preparation. So it's like, this is done. I'm just going home. I'm here for the banana bread and I'm going home. But, I'm going to persevere here. So what we have is a couple of common themes have emerged. And so I'll try and share with them what we've seen as successful. And I'm a lawyer, so we like to put things in categories. The categories that I have are your team, your intellectual property, your corporate structure, and understanding the economic opportunity. So we'll start with the team. So the one thing that we realize is more than any factor, the strength, experience, and relationships of the team that you put together will define your path to success. You have to develop a team that complements your own strengths and weaknesses, and then you need to give them the latitude and the authority to perform. An example of that is, if a lot of you are in academia and you're very good at the technology component of it, you want to think about who would complement you. It could be a person that's really good in the financial aspects, somebody who can navigate kind of the business field and figure out when is it the right time for you to potentially seek funding and what kind of funding should you seek. And there are a lot of people out in this community and in Europe as well that can help you with that. You don't have to have that person in your day-to-day management team, but it would be helpful to have that person in your ecosystem, whether it's a board or an advisor of some sort. In addition to that, we've worked with Dr. Morris on some of this, is sometimes it's great to put together a compelling scientific advisory board. We're finding a lot of startup companies, even before they decide whether they want to have a go or no-go decision as to whether they want to proceed with the company, pulling together a group of experts to advise them, to let them know whether this therapeutic or whatever it is they're developing is something that there's an opening in the market. And more so when you get these credible members onto your scientific advisory board, it also wows the investors. These people have a lot of credibility. And these are people who could potentially help shape your regulatory path and how you define your clinical trials, how sometimes we've seen this in a lot of life science companies, you pivot. You start off as one thing, and in a couple of years, you're something completely different. And these are people that can help you navigate through those paths. The other aspect of it is as you build out the team, it's not just the financial people and the technological people that you need to have. You also need to build out other areas of talent. You might bring people in as employees, consultants, or board members. But our advice is when you start small, 2 to 4 people. Start off a company. You get to your first point where you're trying to raise funds, get the money, then start building your team. But put together a team that the team is critical in developing the company. But at the same time, as much as team is critical and you're leading this charge, you also need to know when to give up control of your company. And it seems like a bad thing to say, giving up control of your company. But it happens in every company's life. And knowing when to hand off control to the next team is also a critical aspect of success. In the academia setting, we see this in 2 different ways. One is you're the person who's come up with this fantastic idea and you're running the company, through the first level of financing. And then an investor comes in and they say, well, we want to put in another CEO, another management after the financing. How are you going to deal with that? Right? These are questions you need to ask yourself. Are you willing to leave your role in the institution and go into the company full time? And if you are you the right person to do it? Right? These are questions you need to ask of yourself. The second way we see it is if you work in like a research lab and you have a postdoc who's super smart, and this person's thesis is going to become the basis of a new company, and you're kind of the head researcher, and now you have this relationship with your postdoc who's going to start the company. You both might be in the company together. How is that relationship going to change? Is it still going to be a mentor-mentee relationship, or is that relationship going to change over time? And how do you address those? And we see that sometimes where the relationships you may have in an institution or an academic institution don't necessarily translate when you're in a company. And so how does that relationship change? And that's why you bring in other people, because you're able to calibrate with the business and to see how you progress. And then, of course, I've got to put in a plug for advisors, right? So when you select a team, the team is just not people within your company, but it's accountants, lawyers, people who do this every day. We've formed, like I said, hundreds of thousands of companies, and we're not reinventing the wheel here. So we're able to give you advice, pick up the phone anytime. And I know at least in our practice and other practices, we're just so happy to hear from people who are trying to form companies. Sometimes you can just get advice and say, this is what I want to do. Is this the right way for me to proceed? And we all just love being part of this community. And so leverage that as much as you can. So that's the team. The second important, and these are in no order, this is just kind of like how my brain works. Intellectual property. You need to understand the process, the timeline, and economics for getting intellectual property out of your institution. So the advice that we give founders are become really good friends with your tech transfer office. Like really know them, take them out to lunch, figure out what you need to do. And then also then use the advisors who've negotiated licenses out of your tech transfer office. And then understand that when you do get a license, that the result is never going to be perfect. They're going to be things that really upset you about a license. They're going to be things that give you a pit in the stomach and you're like, I can't believe I let that provision stay in there. And it's because these licenses are not going to be perfect, and they're going to be renegotiated over time. Every financing seems to be an opportunity to say, well, should we go back and try and renegotiate this term? An investor may come in and say, I don't like this license at all, we need to renegotiate. So just, you got to be comfortable with being uncomfortable with your core license that comes in. And that's just your first license. The other thing that you've got to think about is, how do you continue to develop the IP in the company without running afoul of the IP assignment provisions with your institution? If you're all affiliated with an institution, there are some, every institution has guidelines for how you assign IP into the institution. Now you have another company. So you've got to think about how you navigate those waters. And so find out what those guidelines are. If you're thinking of starting a company, please go find out what the guidelines are in Copenhagen, what the guidelines are here, and make sure that you're talking to someone to figure out how you get the IP in. And getting off, kind of leveraging Dr. Morris's point, is patent, and patent portfolio is important, but be prudent in how much time and money you spend on patenting an idea. There are sometimes significantly diminishing returns for a startup company. Talk to someone about your patent strategy and what it is that you want to patent. Just don't patent everything under the sun. There is a point at which it doesn't matter. So you've got to talk to, like he said, maybe the patent litigators, you talk to patent prosecutors, and figure out what is it that you're trying to own, and be sure that you own it. So there's a little bit of, you just got to temper that a little. So in the same note, corporate structure. Every year, there is a time, and I don't know when it is, sometimes it's before Thanksgiving, we always get these calls from people that say, I've read something in a publication, and this is how I want to restructure my company. Can I get an LLC with a C Corp here, which maybe has a JV here, and another LLC here, and I want to put the IP in one company. I want to put, the investments go into this, and I think this is the best tax structure, and this is going to be fantastic. Can you do this for us? Sure, we can, but why, right? So the question is, we know someone published this and it's making its rounds, to everyone. The people are emailing each other. It's on Facebook or LinkedIn, and everybody wants the coolest new thing. But the question is, complicating your corporate structure isn't necessarily giving you a return, right? So when you talk to lawyers, try to keep it simple. It doesn't mean a complicated structure isn't for you, but 90% of the time, I'll even go 95% of the time, you don't need it. And what will wind up happening is a VC will come in and say, undo that entire thing. Like, we're not going to invest in that. Go back to your simple structure. So try not to overcomplicate what it is. If you're in the United States, we'd say US Delaware C Corporation. The way to get formed, everybody knows how it is. It's in law school, when you're studying for the law exam, the bar exam, they tell you, be sheep. So be sheep. It's like, look like everybody else, then you get funded like everybody else. Like, let's not try and change that. In Europe, would follow local jurisdictions and the conventions over there as to what kind of entity you need to form. So I would say try to be as simple as you can. If you come to the United States, it'll wind up becoming a Delaware C Corp. And lastly, in my mind, it was understand the investment opportunity. Because no matter what a great therapeutic or what you have, if you can't translate that into what return it's going to give people, you can't get funded. Things to think about there, you need to know the economics. So you want to think about how long is it going to take you to get to the market, and what are the costs to get to the market? Does your solution compare with other similar opportunities, or is it still a very early stage product? You want to think about possible market size. How many people are affected by the disease you're curing? For example, is it an indication that affects a large population? There are alternative ways to create value for life science companies, and it doesn't need to be a large patient population. Some things that companies that are looking today are to kind of leverage regulatory opportunities and saying, well, maybe we want to go after an orphan drug status. Or maybe we want to say, we shorten the regulatory approval pathway by trying to get an accelerated approval process from the FDA. And those things, the orphan drug addresses one problem, and the accelerated approval kind of addresses another. Those are things, sometimes it's better to go after a smaller indication and then a blockbuster. So you want to think about that. And this all goes back again to team. It depends on what you're trying to solve, and talk to the experts and try and figure out how to draw these people in to give you the advice. And we have a community that does that. And so going back again to this comment that decimated my entire speech, which it is, a lot of it is luck, and a lot of it is timing. But as that expression goes, luck is what happens when preparation meets opportunity. And so if you can prepare this way, I think you can be exceedingly lucky going forwards. Thank you very much, and best of luck.
Well, very exciting how to put the ideas together, how to get value into what you have. But now comes a very important aspect that I don't know about you, but I feel very intimidated. How do we deal with the government? How do we deal with the FDA? A few years ago, I had the privilege of meeting Dr. Basil Perry and talking about all this process, and I found that he has so much a way to display this pathway that is so encouraging for all of us to be part of an entrepreneurship effort. Dr. Basil Perry is a Chief Medical Officer for Pediatric and Special Population on the U.S. Food and Drug Administration. As Chief Medical Officer for Pediatric and the FDA, Dr. Basil provides executive and clinical leadership on center policy and initiatives associated with medical devices intended for the use of pediatric and special population. He has been a force of improvement, and most important, make the pathway easier for researchers to understand how to link FDA and the NIH. With no more time, Dr. Basil Perez, time is yours.
Thank you very much, Waldo. I appreciate it. Just want to make sure everybody can hear me over there.
We can hear you well in Copenhagen.
Great, thank you very much. Since I am coming to you from DC, I wanted to open with some breaking news that we have. I know everybody thinks of the FDA as the Food and Drug Administration, but I wanted to let you know that we just received word that we are going to change the name of the FDA to reflect the innovation that is happening in devices and the great leadership that is happening at our Center for Devices and Radiological Health. So we have heard from the White House that the FDA will now become the Food and Devices Administration. I'm hoping everybody agrees with that. It was a joke, but we're still the Food and Drug Administration. But I just wanted to clarify that there is an entire center for devices and radiological health, and that is the where my primary appointment is. And I wanted to provide some insights from our Center for Devices that hopefully reflects on some of the points that the speakers have already brought up. I think one of the topics that I'd like to maybe contextualize my comments around is that issue of innovation and safety, and also try to address maybe on the fly here some of the things that the speakers have already mentioned. I think Dr. Schneeberger mentioned that the small innovators need to be supported towards developing technologies that can make a difference across medicine, but especially here and for this conference, across the areas of transplant. And I think I heard also from Dr. Morris and Ms. Deepa that the key issues here are really starting small and recognizing what your timelines are. So perhaps I'll try to provide some insights of where we have been working to help all of those areas. And I know I only have a few minutes, so I'll try to limit these points, and I'm happy to answer questions at the end if we have time. But getting back to that point of innovation and safety, I think many people think about innovation as an area where there's potential risk. There's a lot of risk and there's really a great deal of risk aversion when we think about developing new technologies, new drugs, but also importantly, new devices. The way that we think about it at CDRH is that innovation and safety really are two sides of the same coin, maybe the yin and the yang of how things should work. And the reason that we believe that is that new technologies, new devices can make medical practice and medical care safer. It provides opportunities to have greater life-saving options for physicians to care for patients. And it provides patients with the opportunities to engage their care in a more meaningful way that makes sense to them. So really, when we think about innovation, it really is a matter of creating more safe options and safe opportunities to enhance care for patients. And part of that, we've developed our medical device safety action plan, where we are really protecting patients and promoting public health. And that medical device safety action plan really highlights a few points that I'd like to share with you. We're really trying to establish a robust medical device patient safety network in the United States. We've been working with the MDIC, which is the Medical Device Innovation Consortium, to develop NEST, that's N-E-S-T, that's our National Evaluation System for Health Technology. You can certainly Google that information so that you can get a little bit more detail on that. I'll leave that point there for right now since there's a great deal there. Another aspect of our medical device safety action plan really is to explore regulatory options that will modernize our regulatory process and continue to provide timely access of new technologies for patients. We want to spur innovation that truly leads to safer medical devices, and we want to advance medical device cybersecurity. As part of all of these aspects, we have gone through a significant restructuring at our Center for Device and Radiological Health. That restructuring is what we're calling kind of our TPLC approach to device safety. And that TPLC stands for the Total Product Lifecycle approach to device safety. As part of our reorganization, we have begun to provide greater cohesion and interaction across our pre-market and post-market offices, and all the activities that those offices do are now far more integrated. And we believe that this change in organization at the center, along with our efforts to change culture at the organization, will hopefully continue to support innovation across the ecosystem that, again, brings safer medical devices, life-saving medical devices to the US public at a faster pace. Allow me now to maybe highlight a couple of programs that perhaps are key to innovation. And I'll start with our early feasibility program. Our early feasibility program really is intended to help the early innovator bring a novel technology to the US and to ensure that patients in the US have access to those novel technologies in a timely manner. And we usually try to say that we would like to have the patients in the US have access to those first in the world. That notion is not necessarily to breed competition. It is merely a metric that we want to continue to work towards and ensure that, again, patients have access here in the US to those novel technologies that can make a difference for them. But the key aspects of the early feasibility program goes back to what some of the earlier speakers had noted, starting small. With the early feasibility program, individuals and sponsors can begin a clinical investigation in the US with a small number of patients. There are many technologies that haven't had any experience in human subjects. And because of that lack of experience in human subjects, many are very hesitant and reticent to be able to take on those types of innovative processes for evidence gathering. There's a great risk sometimes with an institution supporting those types of technologies. So what we have said is that if we can work with the sponsors to develop a small subject trial, perhaps just a few, 5, 10 patients, to begin to understand for those specific patients that are at highest risk, where there are no other significant opportunities that can make a difference in their care, that we would be able to approve a small trial to begin to understand whether the device and the technology can make a difference. As we begin to learn more about that, as the physicians begin to gain more experience, as the teams, all the support teams, the nursing staff, the perfusionists, and everyone that takes care of those patients begins to learn more around that technology, we can continue to provide that access to additional patients. And throughout that process, we can get to the point of having a pivotal trial once we have been able to assess that early evidence. The early evidence will let us know whether there are safety concerns, whether there are major issues or problems occurring. And if there are issues and concerns, we can work with sponsors to be able to mitigate those issues and hopefully correct for those issues, especially with respect to technologies. I'd like to maybe pause here and make a point that technology innovation in drugs and technology innovation in devices are very different in this sense, right? Sometimes we might say that a drug development can take 7 to 10 years. You have clear phases of your trials, large numbers of patients, many times, sometimes small, but many times large. With devices, technology innovation can be extremely iterative. They can happen very quickly, and those types of iterations can make a significant difference for improving safety and improving the effectiveness of a device. And we want to continue to support those methods that will help to ensure, again, effectiveness, and most importantly, ensure safety for the patients that are involved with those devices. So that's basically a general overview of our early feasibility program. Let me also give you just a broader, quick overview of our digital health program. Our digital health program is a clear reflection. It's been in development for many years, despite the fact that many may have just heard of some of our recent releases with respect to guidance documents and our white papers on digital health. But we recognize that digital technologies have the potential to revolutionize medical care. And it also has the potential in a very unique way to engage patients more effectively in their care. And because of that, our digital health program has taken on the issues around what does it mean for software to exist as a medical device? And that software as a medical device can also lead to, obviously, artificial intelligence, where you have both discrete algorithms that are closed algorithms that will always, you know what the inputs are and you know what the outputs are. But as that evolves, we have the move towards machine learning and deep learning. When we get to the points of machine learning and deep learning, we're getting to a very novel issues with respect to regulating medical devices. We want to continue to support the innovation in the field, but we also need to ensure that these devices and these algorithms and the artificial intelligence that is being used in devices is continuing to be safe. And when we start to get to that point where we have open algorithms, where algorithms and or devices per se are learning on their own, we need to ensure that those devices are learning from discrete sets of information that are accepted as gold standard in terms of definitions. And as they continue to learn from real world evidence, the broader medical community and EHRs, we need to understand that those algorithms are continuing to put out the right information to ensure that safe medical practice continues for patients. So those are some of the areas that we're working on with respect to our digital health program. And I'd like to close with highlighting issues around pediatrics. Despite all the programs that we have put into place to help support innovation, to recognize the needs of industry, to ensure that patient safety is primary, we still continue to have underservice with respect to pediatric patients and innovation. We know that innovation can make a significant difference in people's lives, but children are people too, and we need to ensure that innovation works for them. Over the last decade, our programs that have supported pediatrics, they continue to yield very few devices that are specifically designed, evaluated, and labeled for children. We have less than 10% of all class 3 or high risk or life-saving devices over the last decade that have been labeled for children younger than 18 years of age. Only 4% have been labeled for children 0 to 2 years of age. So especially that neonatal population that has the highest risk of death from congenital issues, these technologies that truly can make a difference for them certainly are not being developed. And we recognize the numerous needs across the ecosystem with respect to investment in these areas. And we are continuing to work with numerous stakeholders to develop a program that really can create a robust medical device ecosystem for children. This framework that I've put forward, the strategic framework, for right now, we've called it SHIP, S-H-I-P, the System of Hospitals for Innovation in Pediatrics. And the key word here is that it's a system. It's not just a network. It's a broader collaborative public-private sector partnership that will hopefully begin to transform traditional business thinking around investment in pediatric medical device development and medical device development more globally. And we believe that if we can get this framework fleshed out with the assistance of numerous collaborators across the ecosystem, that hopefully within the next few years, we will have a significant improvement with respect to devices that are being, again, developed, designed, evaluated specifically for the complex needs of children. If we can work collaboratively together, I believe that we can get to a point where the entire ecosystem works not just for adults, but works for adults and children, and actually advances and facilitates medical device technology innovation for all Americans. So with that, I will conclude and say thank you once again to Waldo and to all the organizers for the invitation, the opportunity to speak. I truly appreciate the opportunity to be able to engage with such amazing thinkers on this topic across the US and across the world. Thank you once again. Look forward to the panel.
Thank you, Dr. Pires. This is Stefan from Copenhagen. This was extremely valuable, and what you addressed, innovative technologies that are coming to the market specifically addresses the field of transplantation. If you would see this Congress here right now, the actual proportion of the innovative devices is eating up more and more of the industry space. So right now we do have a lot of new devices coming or being at the market already. So this is specifically important. Thank you for making this important contribution. Also, I'd like to just briefly feedback to Dr. Rich. Thank you for pointing out the hurdles that are on the way and the options that we might have with orphan drug approval and expedited approval through the FDA and that channel that is also being addressed, as we know, by some of the companies who try to bring their products to the market more rapidly. Thanks, Randy, for that introduction of the subject. Also, however, for pointing out the many obstacles and hurdles that are on the way if you want to develop a product and eventually bring it to the market successfully. You make it sound difficult and grim, of course, with a 2% success rate. So I hope that we can balance this out now with 2 examples of people and companies who are well on their way to hopefully successfully enter the market or who are on the market already represented. I'd like to start with Dr. Edward Chong. Eddie has vast experience in, with medical companies, over 25 years in drug developments, both in small but also in large industry. He now serves as a CMO at Vitaris. He's the lead clinical and regulatory team, and he works with the FDA, so I'm sure he's very, very familiar with what was just said. Eddie, we're looking forward to hear your story.
Yep. Thank you, Stefan. Yes, my name is Eddie Chong. I'm the Chief Medical Officer of Vitaris. We're a small, almost virtual company based in Vancouver in Canada. And what I'm planning to do over the next 10 minutes is to describe the course that we undertook from the time we started the company to where we are today. And over this course, over the last 3-4 years that we started the company, I will touch on many of the themes and points that the other speakers have made, in the sense that you need to have a good asset, a well-understood asset. You need to have a clear idea of where you want to place, where you want to develop your drug or asset for, preferably an indication with a high unmet medical need. You need to have a clear regulatory pathway, and then you need to have a team around you to implement the development plan. And that's also probably the most crucial point that I would like to make. So Vitaris was started in 2016. We're a bit unusual in the sense that we did not come about from a university laboratory. The company started when our CEO and founder of the company persuaded his company to spin out an asset, in this case, clazakizumab, an IL-6 antibody. And this asset was sitting on the shelf in his previous company, gathering dust. And this asset had been developed by his original company and by another big pharmaceutical company up to the end of phase 2 with positive results and with a lot of data, human data, and also a complete non-clinical package. So all the toxicology studies have been done. The manufacturing has almost been completed. So we had this asset that was sitting on the shelf and ready to find an indication. So that's how the company was started. We started in 2016, and then today we have just started our phase 3 study, our pivotal study. So don't get too excited. This is incredible. If you look at how long it takes to get a drug from the bench to a phase 3, it will take you many years, not in the time span that we've been around. The first year of the company's life, we actually we went into a different direction. We got our feet under the desk. So we actually started in this indication the last 3 years. And the indication that we chose was for the treatment of chronic active antibody-mediated rejection in kidney transplant recipients. As Stefan was saying earlier, treatment of acute rejection in kidney transplant is well taken care of with the current immunosuppressants that we have. And the biggest problem that the transplant community finds now is how do you preserve graft survival long term. And the chronic antibody-mediated rejection is now recognized to be the leading cause of graft failure. It's well understood in kidney transplant, but it's also becoming recognized in other solid organ transplant. And there is no effective or proven therapy for this condition. So what we did was we had to design a clinical program that was expedited, convince the FDA and other regulatory agencies to accept our program so that they could give us accelerated approval if we are successful using a surrogate endpoint instead of a clinical benefit endpoint. And one of the issues that Stefan mentioned is that if you want to develop a drug to prevent chronic rejection, these studies, if you use the clinical benefit endpoint of allograft survival or allograft loss, this will take you many years and a large number of patients. So the way to get around this is to design a program using what we call a surrogate endpoint for expedited or accelerated approval in the US or conditional approval in Europe. So that's what we did. We spent quite a few months, or nearly a year actually, working on this and trying to convince the FDA and the CHMP of what we were planning to do. So there is a lot of data out there that shows that renal function is closely associated with graft survival or graft loss. But the problem we had with the data out in the literature is that there is no quantitative relationship, no quantitative data, or data that shows the quantitative relationship between, for example, eGFR decline and the risk of graft loss in the next few months or years. So for example, if you have a 1 mil decline in eGFR, what does that translate in terms of the risk of subsequent graft loss? And without that information, you will not be able to design a trial in terms of powering your trial, how many patients and how long should this study last, etc. So that's what we had to do. This was a curveball thrown to us by the FDA, so we had to do some modeling exercise that took us about 9 months. So anyway, we were successful in persuading the FDA and the CHMP with this modeling that we did, and they agreed that not only could we use this surrogate endpoint, this eGFR endpoint, which is not validated, because unlike other surrogate endpoints like blood pressure, blood glucose, lipids, those are validated surrogate endpoints that you could use to develop an anti-diabetic drug or lipid-lowering drug, etc. But in the field of transplantation, there is no validated surrogate endpoints. You hear a lot of people talking about using biopsy markers or DSAs, but these are not validated, or even not even validated, but even the data trying to support this endpoints as likely to predict clinical benefit. That's the criteria that you have to satisfy the FDA, that whatever surrogate endpoint you're going to use for accelerated approval is likely to predict clinical benefit. There's no, if you look at the literature, it's very confusing in terms of using some of these other surrogate endpoints. So that is the process that we went through with the FDA. And in the same time, we had to raise money as well. We started off with a small group of investors that got us going. And we were fortunate in persuading one of the big biopharmaceutical companies, CSL Behring, to be our corporate partner. And in essence, they're funding our research. And obviously, if we are successful, they will see the benefits of their investment in us. So that's where we are. We are now starting our phase 3 program. We have the sites have been initiated in the US, in Europe, and in Australia. We have coming back to the team, and some of the previous speakers about trying to start off with a small team. You'll be surprised that in Vitaris, we only have 4 employees in the company. The company is headquarters in Vancouver. I'm the only person working in Vancouver. I have a colleague who works in BC, further up north. One of our other employees, the CEO, lives in Seattle. He works out of his home in Seattle. Another employee works in Sydney, Australia. But we obviously have a group of consultants and contractors, about 20 contractors and independent consultants working with us. And obviously, we will have to use a lot of CROs as well to implement our phase 3 program. So that's basically what we've been through with Vitaris. In the meantime, we've got orphan drug designation from the FDA, and that will give us some regulatory benefits as well, and financial benefits. So those are the things that we have achieved in the short time span that we have, that Vitaris have started. Okay, thank you.
Thank you, Eddie. That's an inspiring story. It's fascinating to hear that there's only one person working in one place, and all the other members of a rather small group, considering that you're in phase 2, is scattered around the globe, basically. I think you addressed a very specific task with chronic antibody-mediated rejection. This is a hard nut to crack. You addressed the need for surrogate endpoints or other mechanisms to avoid establishing a formal endpoint first. And it's good to hear that the authorities, the FDA, but also EMEA are responding to the need, because the field is very hungry for that innovation. We need it desperately. and the time to establish the more formal endpoints is too long, as it seems from a clinical perspective. So it's good to hear that the authorities are opening up, they're working with you apparently, and that allows companies such as yours to advance and move forward with that. So thanks for sharing that experience. With respect to time, I think we have to move on. Dr. John Piano, welcome you here. It's a privilege to have you here and to hear your story. You started as a corporate lawyer. That's not an easy job either. And then you left that job to found a company that is now successfully on the market. Transplant Connect was founded in 2003. John, we're much looking forward to hear your story.
Great. Thank you, Stefan. I appreciate being here. You know, we heard so far that it's about timing, it's about luck, it's about complying with regulations, and those are all absolutely true. Without those aspects, no entrepreneur would make it to the next stage or be successful. And obviously, within the context of donation and transplantation, there is a tremendous deal of rigor that's involved. You are all extremely smart people, extremely well-versed in your expertise. And so there's a very high standard that applies in this field. It's not like developing, we're obviously talking about software here. It's not like going out and developing a software application, an app for your phone that a bunch of, you know, you're going to sell to a bunch of teenage kids to take photos of themselves that disappear after 3 seconds, right? It's a completely different marketplace. And there's a whole complex ball of factors that I personally have dealt with, and I'm going to endeavor to share some of them with you in the next 9 minutes here. So focus in on, I started the company after having had some experiences, personal friends, losing loved ones, waiting for organs. I had no connection with organ donation or transplantation whatsoever prior to that. I had been a corporate lawyer, I had been in media and the film business, I had worked in venture-backed internet companies as well, in leadership positions, and frankly wasn't really all that satisfied or fulfilled. And so when I learned about donation and transplantation, and frankly the organ shortage, it was as though I was at the time in my life, and it really created a passion in me, and that's really, I think, what I want to talk about a bit is passion, because you got to have all these other aspects, but if you don't have passion, to be a successful entrepreneur is extremely difficult. The hurdles that you have to get over, the challenges that you have to survive, the disasters that you somehow have to come back from as an entrepreneur, if you don't have the passion to carry you through, you're probably going to hang it up and quit. And that's perhaps one of the reasons why, 98, 99% of all startups fail, is that people don't necessarily have the passion to persevere through the tough times. You know, for us, it started out as I learned about donation and transplantation and the organ shortage. I became fascinated with it. Again, unique time in my life, so the timing was there. I did a whole bunch of research and studying. I spent about a year of my life coming to conferences and researching on my own, and came across a paper back, this was in, my God, 2001. I still remember the name of the author, Edward Guadagnoli. Maybe some of you know him. He's a Harvard health policy PhD, and it was about the pool of donors in the United States, and the true medically suitable pool of donors, and was the current process for organ donation and transplantation effectively capturing that medically suitable pool. And the analysis, he ran through a whole mathematical analysis and very rigorous statistical approach to essentially ascertaining the answer is no. That there's a lot of potential organ donors and donor organs being left and not being carried through for transplant. And then he talked about the various reasons for that, and one of the key reasons was breakdown in communications and processes and documentation, and just the slow delay that really was inherent to the process. And that triggered something in me. It was intellectual, yes, but it triggered a passion in me to say, wow, this totally makes sense, and I understand. I had known nurses in the intensive care unit, and I spoke with them, and I understood, wow, there's a breakdown that's occurring here. So what we did, raised some money, and built out, our initial plan was to actually create interfaces into the hospital EMR systems to automate the process of donor referrals from a hospital to the organ recovery agencies in the United States. And we had been successful in going through layers and layers of scrutiny with Kaiser Permanente, which is a very large hospital network in the United States. We got approvals. We started building out this interface with their electronic medical record system. Basically spent all the money that I had raised. I had stopped drawing a salary, I think, about 4 months, and I was just working, hoping, praying to God that we were going to make this interface work. And the tests worked. We were able to successfully push data from the EMR into our system, and it all looked great. And then one day I got this horrendous call from the CEO of Kaiser saying, we have to pull the plug on this project. It's not going to work for some internal reasons that we don't need to get into. So here I was, basically out of money, spent about 8.5, 9 months, and we didn't have a product, and we had no potential for anything in the near future. And that's when I think perhaps 98, 99% of people would say, okay, I'm done. Let me go back to a law firm or go back to a job and learn to pay the bills. But honestly, the passion inside of me said, no, there's something not to be given up here. And we were able to pivot, which is another key term in the field of entrepreneurship, right? Often the first idea may not be right at that time, so you pivot to something else. And we were able to do that, and we focused on the organ donation systems, building out full electronic medical records for organ donation, not just wait list management and not just capturing enough data to allocate organs, but actually managing donor cases and capturing that data and leveraging that data for better decisions. And we were successful in that. And today, about 80% of the United States operates on our platform, all of Australia, most of Canada, and we're hoping that we will be successful in coming to Europe. But the key point, I think, is, so here we are today, having launched that. We've now launched our transplant center system as well for patient management. But inside of me, that passion for the original idea of automating the donor referral process and creating those interfaces, I never gave up on it. I knew that we were maybe a little bit ahead of ourselves back in 2004 when we first tried it, but and maybe it wasn't fully intellectualized and we didn't run all sorts of studies. It was more a gut instinct that this is going to work. And so we went back to it. And after an immense amount of work, and one of my colleagues here, Wade, who really has done so much of the work with his team for us, last year, after about 4 years of persevering, we successfully launched that interface in Texas. And we interfaced with the Epic EMR system to automate the donor referral process. And the beauty of having carried through, I mean, I feel such a passion for having done what we did, because not only does that interface successfully save time and eliminate phone calls and streamline processes, but that initial paper by Edward Guadagnoli that persuaded me back in 2001 or 2 that this is something I should do, I think he was right. We launched that interface, and after 6 months of use, comparing it to the prior 6-month period where they had no interface and it was all a phone-based donor referral process, so immense amount of time was saved, about 40 or 50 hours per month was saved at the hospital level and at the organ recovery agency level. But the donor, the numbers for donors in that hospital, the year prior with the phone-based process, this hospital had 1 donor over that 6-month period, and that 1 donor yielded 3 organs for transplant. So after we launched our interface, that same 6-month window with our interface the year later, they generated 7 organ donors with 18 organs that were successfully transplanted. And I guess what I'm trying to say is, had I not continued to embrace the passion for that original idea, a lot of people I might have consulted with would have said, John, you're an idiot, give it up, move on, just focus on the organ donor systems and focus on the transplant center systems. But to have, sometimes, being an entrepreneur means going with your gut, and you don't have all the data in front of you, and you just have to persevere. And that's a big part of our story. The reason Transplant Connect today, I think, is where we are, is as successful as we are. We have a lot of brilliant people, immensely brilliant people, far more brilliant than me, who work for me. But I think it's following that gut instinct and that passion. And even if you're working in a highly complex field, if you don't have that passion, I really wonder how you can truly be successful. I mean, all of you being surgeons or coordinators, if you don't have the passion for what you do, life is too short. You should be doing something else. And that really applies to entrepreneurship, because you go through a lot of pain as an entrepreneur, as I'm sure Eddie can attest. And any successful entrepreneur will tell you, you hit rock bottom before you then eventually dig yourself up. And that certainly has been my experience. So not a very technical talk, but you know, it's important to realize all aspects of our efforts. So and that's really what I wanted to share with you guys today. Thanks.
Thank you, John. Not technical, but inspiring. Thank you for sharing that story. It's a wonderful thing to hear. Would you have known about all the obstacles and the pain that you're getting yourself into? Would you have done it?
You know, I think, to be honest with you, my preparation, having been a corporate lawyer, prepared me for a lot of pain and a lot of obstacles. So that helped me to endure, I think, to be very honest with you. And I was very patient. I knew that it wasn't going to happen overnight, and somehow I was able to persevere. But yeah, and Eddie, you're talking about being patient. You have to be patient, because things aren't going to just land in your lap. I guess sometimes they do, but most of the time they don't.
Well, thanks, John. Now I'm giving the word back to Randy. I see you at the podium already. I hope you hear us well. I hope you enjoyed the stories as much as we did here.
Yes, I must admit, I am very, very impressed with the technical support staff. Things have gone so smoothly from Stanford to Washington, D.C. to Copenhagen. I really didn't think this was going to work. But you know that I'm a regular cautious individual after you heard my talk. I have the great pleasure of introducing Dr. Richard Miller, who is President and CEO of Corvus Pharmaceuticals, which is an oncology company. Richard is a true professional entrepreneur. I remember, I think I was a medical student when I sat down with him with the blueprint of his offices for his first biotech, body-based company called Biotherapy Systems, I believe, which morphed into IDEC. IDEC then joined Biogen. Then he formed Pharmacyclics. Then he formed Principia. Now he's formed Corvus. So he is also an adjunct professor of oncology at Stanford. Somebody I have gained a lot of benefit from sounding for over the years. He'll give me his totally unvarnished opinion about my crazy ideas. And I hope he'll educate you as well.
Thanks. Thank you, Randy, and thank you, Dr. Concepcion, for inviting me to this very prestigious meeting. Randy already mentioned that I've known him for a long time, actually, when he was a medical student. And I like to think I taught him a few things along the way, but he probably taught me more. We've heard throughout this symposium that luck and timing are important, and they are, no doubt about that. But I still am a firm believer that the most important ingredients are great science and great medicine. And with that, I'd like to share with you in the 10 minutes or so I have some of my experiences, because I think they set perhaps good examples. One of the really challenging things for academicians is they come up with a discovery or observation in the laboratory. They think it has some clinical utility or some application. They're too advanced for NIH grants. They're too late for the early funding. And they're too early for the late funding. They go to Big Pharma says, oh no, it's too early. So they're in this, what we call valley of death. So the valley of death is the period of time in between your discovery, usually in the academic laboratory, and gee, how do I get to proof of concept? Proof of concept could be animal studies, laboratory studies, more importantly with the therapeutic, early phase 1 clinical data showing some pharmacodynamic or clinical benefit in a small group of patients. So how do we bridge this valley of death? This is the biggest challenge an entrepreneur faces, especially these days when research is multidisciplinary and quite expensive. There's so many great technologies out there, we can't do them all. So we need to rely on vendors and contractors and collaborations to get things done. So how do we bridge this gap? Well, you can go to your uncle and ask for $50,000. I did that, that didn't work. You can go to your relatives. You can go to the incubator at your local university and get $100,000 if you beg and borrow and write a 100-page grant. That's too long and difficult. So you really need to, at least for therapeutic products, you need to secure a lot of money. And a lot of money, I mean tens of millions of dollars. So what does it take to do that, to bridge that valley of death? Well, I think it's pretty obvious to the people in this room and the people at Stanford and at the ESOT in Copenhagen. If you're running laboratory programs, you need a good idea. Something really, really good that's exciting and people around you are excited about. You need to work in a hot area, what I call a hot area, exciting area, because investors usually chase things that are hot. And even if it's not hot, you got to make it sound hot. Today, I learned from transplanters that antibodies to donor organs is really a major hot topic. So that's something I would be thinking about and already am as I sit here today. What do I mean by hot topics? Today, CRISPR's hot, bispecific antibodies are hot, CAR T is hot. 3 years ago, it was immuno-oncology. That's sort of cooled off, but it'll get hot again. Success and failure pushes these cycles. So work in a hot area, be a leader. Investors like to work with leaders. Strong IP goes without saying, an idea that's clearly patentable. And a development plan that is easy to measure and has clear inflection points. What do I mean by that? Well, if you're going to work on a drug that you can easily measure we'd love to measure its survival, but that's hard, and that takes a long time. But are there pharmacodynamic markers, or what we now call biomarkers, that really are going to tell you in animals and in people that the drug is doing what you think it is doing? That's really important. Because as you try to navigate through that valley of death, these are the things that really will drive financings. And I think as Dr. Morris answered, I'm not sure I would say you need the product label ready, but you certainly need to know how you define success in a phase 1 study. Can you tell your investors, look, if I can do this, this will be great, and doctors will want that. Patients will want that. It will address an unmet need. Design of clinical trials and animal efficacy studies that give you these clear endpoints, pharmacodynamic or efficacy endpoints, is critical for driving value. So what does it cost to get all that? So I thought you'd be interested in this. A small molecule, easier to manufacture. If you want to make a GMP drug to test in IND-enabling toxicology studies, maybe $500,000 to $750,000. That's not too bad. Vendors will do that for you. They'll even work out the synthesis for you. Many companies around the world will do that for you. The IND-enabling studies, tox studies, and filing an IND is probably about another $1.25 to $1.5 million. So pretty much for under $2 million, you can get your drug made and get in the clinic. That's not too bad. It's not $2 million isn't hanging around on the street corners, but it's not insurmountable. Now, antibodies and proteins, different game. Proteins are more expensive, antibodies, for example, because the manufacturing of GMP antibody to get into the clinic is $4, $5, $6 million, depending upon the productivity of the clone that's secreting the antibody. So, and then the tox studies are another million. So small molecule you can get in the clinic for under 2 million, I would say. A protein antibody molecule, for example, probably $6-7 million. And they both have advantages and disadvantages. The advantage of an antibody is, even though biosimilars are here, they have not really substantially eaten into revenue in the US as of yet. So you have a longer term revenue stream. So let me give you an example of a couple of drugs that exemplify this really well. So our team at Pharmacyclics had the idea, really something we had read in the literature. Scientists had made a covalent drug to EGFR, epidermal growth factor receptors, kinase. There are 300 kinases in the body. And there are about 30 kinases that have, well, actually about 100, but 30 that have accessible sulfhydryl groups, cysteine residues in the ATP binding pocket. Now we'll get a little technical. So these workers had shown that you could make a drug that would look like ATP, get into that pocket, react covalently with the sulfhydryl group, and block the kinase activity. So we said, okay, well, we were oncologists. We don't know anything else but oncology. So we said, well, there's a kinase called BTK, Bruton's tyrosine kinase, which turns out to be one of these kinases that has a cysteine in that ATP binding pocket. And so we were able to make a drug to that pocket. And this is where it was really kind of a clever idea. We knew that would work. Well, how did we know that? Because Dr. Bruton in the 1950s described a disease in boys called X-linked agammaglobulinemia. The BTK is on the X chromosome. These kids have a mutation. They don't make BTK. They don't make a functional BTK. And their B cells don't develop. They're agammaglobulinemic and their B cells don't proliferate. So we said, well, maybe this will be a good drug for lymphoma, for B-cell lymphoma in people who don't have BTK mutations, but perhaps BTK is driving this tumor. And we had some in vitro evidence to suggest that that was true. So we made this BTK inhibitor, and we had some key assets that I just discussed. Number one, we could test its occupancy in the target. So we could give this drug to an animal or to a human being. I could take a blood sample and I could measure the drug sitting in that ATP binding pocket. Perfect pharmacodynamic measurement. Helps me with dosing and safety and function and all that. Secondly, we are able, we could do this in mice, but mice are terrible models for cancer. And I share that you have the same feelings about transplantation. We went to, instead of mouse models, we actually said, well, dogs get B-cell lymphoma. Pet dogs, companion dogs, get B-cell lymphomas that are very similar histologically to human B-cell lymphomas. You'd have a hard time distinguishing them under the microscope. So we treated about 10 dogs with B-cell lymphoma with this BTK inhibitor, which is now known as ibrutinib, by the way, or Imbruvica. It's actually the 5th largest selling cancer drug, but moving up quickly. So Imbruvica, or ibrutinib, in these dogs, of course, we didn't know the dosing or anything like that. Dogs are a little bit hard to get CT scans on. But we saw about half of the dogs respond, and that was actually published. And so this was fantastic. You know the drug gets to the target. You can measure it on a blood sample. You see activity in dogs. You go into the clinic. Sure enough, you take patients in your clinical trial. Again, here's where good phase 1 design, where you can do things that you can measure. We took lymphoma patients that had circulating tumor cells, chronic lymphocytic leukemia, for example. So all you had to do is take a blood sample, and you can measure the drug in the tumor. You could also see whether or not the blood counts changed, go down. Sure enough, that happened. In a phase 1 study, it was clear the drug was working. Those results were published. Eventually, a randomized phase 3 trial was done. in a related disease called mantle cell lymphoma, got accelerated approval because mantle cell lymphoma, deadly disease, deadly B-cell lymphoma. And it was approved by the FDA, accelerated approval in December of 2013. At the ASH meeting in 2013, American Society of Hematology, which is in December, there were over 100 abstracts on that drug. That's what I mean about science and medicine. That's what I also mean about collaboration. The reason I came here to talk today, as much as I love Randy, I really want to begin to collaborate with transplanters because I think there's a lot of similarities between cancer and graft rejection. Serious illnesses, unmet need, great scientific underpinning, ready to take the next leap. So that was a BTK inhibitor. The continuation of that story is that drug right after the phase 3 trials, Johnson & Johnson did a deal with us for $1 billion. Global rights were shared. And a year after that, 2015, AbbVie bought the company for $21.7 billion, and that's for 50% of the drug, because half the drug was already licensed. That's a $42 billion value for one drug. So that's why when Randy says 2% of the companies succeed, he might be right about that number. I'd have to check it. Sounds a little low to me, but that 2% pays really well. And investors invest in biotech, not because they're losing money, but because over the last 20 years, they're making money across the board. Even though there might be more losers than winners, they're getting a great return on investment. That's why they do it. Otherwise, they wouldn't do it. Now, my company, Corvus, we said, hey, that's a pretty good plan. Let's try to do that again. So it turns out that T cells have a similar kinase called ITK that's involved in T cell function. It's involved in cytotoxic T cell function, proliferation, rejection, things like that. I'm not sure there's much known about its role in rejection, actually. So that's a drug. We tested it in dogs. It's, again, covalent drug to ITK, not BTK. We tested it in dogs with T cell lymphoma. Humans and dogs get T cell lymphomas as well. Sure enough, dogs responded. We said, wow, that's great. Now the drug just entered human clinical trials in patients with T cell lymphomas. Studies open US, Australia, and South Korea. A lot of T cell lymphoma in Asia, so we were interested in having a presence in Asia. Again, one of my purposes is I'm really interested in working with Randy and Dr. Concepcion and anybody because we're not very smart about transplantation. And so one of the other really important things I think about starting a company is you have your technology, and you need to focus, but investors really love it when you say, you know what, if I can show it works here, there's an opportunity that I can extend it beyond that. They love that. They love to be surprised. So do I. So basically, I would say that I would take luck any day of the week, and I'll take timing. But unfortunately, hard work and good science and good medicine is really crucial for this. It's if we're going to start companies where we really need to do research, discover drugs or technologies and develop them, you have to start with a great team. I agree with that, but it also takes a lot of people and a lot of money. So it takes a great story to persuade investors that put some money in now, we'll have another inflection point and another one and another one, and we'll keep building value. And even though it takes 10 years to make a drug, there's plenty of points along the way where you can step off this train and make money. Thank you very much. Great talking with you.
It's exciting that we're starting a dialogue on different entities that can make a difference and take care of patients. Just to give a number out there, if everybody of you probably know this, but just to give a little information, 25% of our list, these patients have received a transplant or are highly sensitized. And these are the patients in which they're not going to get a transplant in 1 year, 2 years, 3 years, 4 years, try 8 to 10 years waiting on the list. If we would have the technology, we had medications that can take care of the problem of antibody-mediated rejection, B-cell biology, and tough T-cell biology, we can make a really progress in the field. This is why inspiring talks like this and projects that come from Europe and United States put together, hopefully we inspire the next generation and create this a purpose of solving problems. It's an honor and a privilege to have the opportunity to introduce Dr. Matthew Rabinowitz, that is a co-founder and chair and board of directors of Natera. If you look at through his CV, so much has done, developing companies, developing technology, most important, making a difference in the field of genetics. He's a consulting professor at Stanford University from 2004 and 2012, and in 2018, he's a visiting faculty at genetic department at Harvard University. Private is also have done significant amount of research with 6 grants with the National Institute of Health. And if you look at his publication, it's all wide variety in highly valued journals. It's an honor to have you here, and we're looking forward to hear about your experience. Thank you for being here.
Thank you very much, Waldo. Good morning, Stanford. Good evening, Copenhagen. When I was about 5 years old, my mother was out, and my father, unsupervised, decided to play mind games on his little kid. And he said to me, "Matt, you seem to have an answer for everything. Tell me, where does space end?" So I thought about this for about 3 days. And anything I imagined as a barrier to space, there had to be something beyond it. And I ended up piled in a heap on our driveway in Johannesburg, South Africa, crying and crying, because I could not imagine a question that I had no way to even conceive of. And my talk is going to be largely about accepting that paradigm. At the same time that entrepreneurs and doctors, especially transplant doctors, are trying to control the world, trying to optimize life, we have to, at the same time, connect to that place of acceptance. And I think it's the most important thing for an entrepreneur to learn. This is not quite the same message as one typically gets in entrepreneurial talks, which talk about, passion, intensity, forcing change. I would say that connecting to that place of what some call dharma, what some call grace, what some people talk about as angels singing or heavenly light, actually makes you a much more effective agent for change in this life. So my first companies were all about optimization, controlling the world. I started a company that was about intelligent merchandising online. We looked at the internet in the late 90s, and we thought there's going to be way too much information for people to sift their way through. And so we built the first versions of profiling of people. So you like these books, you like these movies, these are the things that you would likely be interested to see next. And it was just the right thing at the right time. The next company was all about optimized location technologies and communication systems, which I started with the 2 guys that architected the GPS system for the US Department of Defense. They were also Stanford professors at the time. And we combined GPS with digital television to create this high bandwidth, high power GPS that worked indoors. So that was a really cool technology, but I was getting to the place in my life where I was tired of working on communication systems and making things go ping on the internet. I thought there must be a more fundamental way to spend my next 10 years working on things that affected life and health and happiness. And it was around that time that my sister, who was 32, had a child with Down syndrome. And it was a surreal thing for a family to go through. The child died after 6 days with all sorts of complications. And I couldn't imagine how in the 21st century, we were fooled by these serum screening tests and we were unable to find something so basic. So I gathered a fledgling team out of Stanford University, and we started to work on new techniques for analyzing single cells and small amounts of DNA, such as in the non-invasive prenatal setting, where you look at cell-free DNA. And we created, to my knowledge, the fastest growing genetic test in the history of this country, which is called Panorama. It's the test where instead of doing an amniocentesis where you stick a needle in a woman's belly and it has all sorts of risk to the pregnancy and it happens pretty late in the pregnancy, you just do a blood draw from the pregnant mother's arm, and you can look at tiny traces of fetal DNA in the mom's blood and detect all sorts of chromosomal structural abnormalities. Now, the reason why that technology worked well, I would say, is because we had a very multidisciplinary approach. Instead of just focusing on the measurements in the lab, we came at this from a statistics and informatics intensive angle. The other techniques that are predominantly out there, besides our test, are looking at shotgun sequencing methods where you measure the amount of DNA, and if you see more DNA on particular chromosomes than you expect, you say, okay, that's aneuploid. But those techniques tend to generate a lot of false positives and false negatives because I see a couple of people nodding in the back. Thank you for your support. They tend to generate, there's a lot of variability in biological samples where you will see more or less DNA in the plasma, which varies from one patient to another, and you can sequence as much as you like. It's a biological bias in a patient that you can't control. So the way we approach the problem is we built this massively multiplexed technique where you could target SNPs, single nucleotide polymorphisms, where there are genetic differences between people. I might have an A at that locus, and Waldo might have a C or a T or a G, and we could get information from targeting these SNPs, which lets us figure out what fragments of DNA, what haplotypes from the parents constituted the DNA of the child. And we developed this maximum likelihood Bayesian optimization technique where you look at literally billions of hypotheses of what could be making up the DNA of the child, and you evaluate each of those hypotheses given the measurement that you've made on the plasma, what you know a priori about the parents, what you know about your noise profile of your amplification and measurement platform, and also what you know from the Human Genome Project, like the HapMap database, which tells you where chromosomes tend to cross over during meiosis. And one of those hypotheses could make sense of your measurements as well as all of your a priori information. And as you evaluate those hypotheses, you're also integrating across all the fractions of fetal DNA to find that fraction of fetal DNA that exactly explains the measurements and the prior information, and that stands up above the noise as the hypothesis that makes sense of the world. So now when you get that hypothesis, you don't just know what's the ploidy state of the fetus, you also know what's the fraction of fetal DNA in the maternal plasma, and you can figure out the confidence in your call, because knowing the precise fraction of fetal DNA, we know what it should look like if there's an aneuploidy. So the result of this technology is we could differentiate the fetal DNA from the maternal DNA, and we could measure all sorts of things with a much more powerful test statistic. We could look at subchromosome structure, like a 22q11 microdeletion, which causes schizophrenia, heart abnormalities, and retardation, that the shotgun methods just couldn't measure reliably. We could look at vanished twins, molar pregnancies, triploids, all sorts of things that you couldn't see, and see that much earlier in the pregnancy at about 9 weeks. So we've had amazing growth at Natera. I think around next year, we'll have about 1 million prenatal samples a year run rate. So it's been pretty phenomenal. And then we turned our attention to other problems that really mattered to us. What else could we solve? And the first problem that we decided to go after was recurrence of cancer. There are 18 million people living with cancer or in remission in the United States, and they are subjected to CT scans, mammograms, a lot of worry, a lot of expensive procedures that are in and of themselves possibly harmful. And we thought, I'm sure we can solve this problem better. So we took the platform that we developed and we tweaked it in various ways to build a customized panel for every patient based on the particular mutations that drive their cancer. And if you go with that approach, you can go down to these very low levels of sensitivity with high specificity. So we were on the cover of Nature recently, showing that we could catch the recurrence of lung cancer well before you see clinical symptoms. And since then, we've been in a lot of the top clinical cancer journals like JAMA Oncology and a bunch of the top journals, showing that we can catch the recurrence of many cancers several months or in some cases years before you see nodules by imaging or clinical symptoms. And the ability to do this with high specificity is wonderful because now you can start to treat patients upon molecular occurrence. You don't need to wait for them to have clinical symptoms where it's often too late. We can treat with particular regimens, with immunotherapies, with surgical interventions, and really change outcomes. That ability to go down to low fractions of DNA is fundamental in the oncology setting. We were going down to about 0.005% tumor-derived DNA. And that's roughly the equivalent of detecting a single molecule of DNA in about 5 mils of plasma. If you look at how much DNA is in 5 mils of plasma, that's about the same as detecting a single blade of grass in about 100,000 soccer fields. And it's just beautiful to see that kind of performance translate into real clinical change. So the next thing we looked at is what are problems that matter to the people we care about? And a lot of the team at Natera knew people or had been directly affected in their own families by renal failure and organ transplant. And because we were able to detect the fraction of foreign DNA so precisely as we developed in the prenatal setting, we thought that must be applicable to look at early rejection of kidneys. So we built this assay, which was derivative of the work that we'd done previously, which had 13,000 SNPs that we would target to detect rejection. And building an assay with 13,000 PCR primes to target SNPs is non-trivial because PCR is a highly sensitive reaction. Every time you thermocycle a reaction, you're replicating, roughly doubling the amplified product. But because it's exponential, you get high sensitivity to primer dimers. And when you have many PCR primers in a single reaction well, you get garbage out of the reaction. It's very noisy, and you get particular regions of the DNA that are represented, and everything else is kind of garbage. So what we did is, again, we sort of leveraged these cloud computing resources, and we built this multiplex PCR assay where every primer that was targeting a particular region was optimized relative to the other 13,000 primers to minimize the cross-reaction products and to have very uniform thermodynamic characteristics, so you have great coverage across the chromosomes. And the result of this is, on replicate samples, we were able to measure the fraction of DNA from the donor with a coefficient of variation of about 1.85%. So if you're at about 0.6% donor-derived DNA, in absolute terms, that's about 0.01% standard deviation on measuring that donor-derived DNA in replicate samples. And that was about 5 times better than the technologies that were around. So again, it was great to see that kind of analytical performance translate into great clinical performance. We did a trial with UCSF, about 300 patients with matched tissue samples, and we showed that we could generate an area under the receiver operating curve for detecting rejection of about 87%, which was substantially better than the technologies that were out there. It was more sensitive than certain, I was talking about cell-free DNA technologies, but when compared to serum creatinine, it was more sensitive by quite a long way, detecting all forms of rejection, including T-cell mediated rejection, which I believe was the first time that had been shown. And also in the monitoring cohort, we were able to see subclinical rejection, I believe for the first time as well, using cell-free DNA technology, with a sensitivity of about 92% and a specificity of about 75%. So we were really excited by this, and that technology Prospera is now available. But still, it's not perfect. And we have imperfect sensitivity, imperfect specificity, and in some cases, imperfect patient outcomes. And so now I go back to the original theme. What do we do about the imperfections of our technology and the imperfections of the process of starting a company? I would say that the ability that doctors have to do their very best and nonetheless accept the hardships of certain outcomes, especially in fields as tough as transplantation, are probably the most important skills to develop as an entrepreneur. Because it is so much harder to take technologies to the market than all the success stories might lead you to believe. And whether you get in touch with that domain of the heart and that area of acceptance through meditation or through conventional religion or through walks in nature or through a sense of source energy, it makes you so much more resilient to accept the challenges of building technologies that are going to do what nobody else has done before. It's a bit like that quote from Hamlet. After so many machinations of trying to control the outcome and trying to control his life, he sort of resigns himself to the hand of God. And he realizes that you can do all you can, but at the end, you just need to accept. And it brings him a moment of peace. And he says, the readiness is all. Let be. So my nuggets for my talk are, I think, pretty obvious, the first set, things you've probably heard before. Obviously, work on problems that you care about personally because you have no idea how long and hard the road is going to be, and you need to be personally motivated. Get into a field that is young and learning and growing because that makes an enormous difference to the rate at which you will grow and the size of your market. Take a multidisciplinary approach to problem solving. Come at problems from many different angles. Use different technologies and different modalities, especially as a transplant doctor and obviously as an entrepreneur. Build great platforms that can be translated to many different opportunities. Work with people who are courageous and caring because life is too short to do anything different. And then the last one is, at the same time that you are putting energy into changing the world and optimizing science from the brain, put the same amount of energy into the heart and learning acceptance, especially in the field of genetics, where we are continually trying to play God. We need to accept that we are really playing man the best way we know how. And we need to put as much energy into acceptance as we put into control, because we are here to learn both. Thank you.
Thank you, Dr. Rabinowitz and Dr. Miller here from Copenhagen. That was two extremely fascinating stories. Randall and Waldo, I think we'll wrap this up and go through the summary statements. If you could bring your speakers to the podium. And if we could start seeing the slides. And I think we're gonna keep this very focused and very short if you can. Get right to the points of what the core message of your experiences is.
Okay, Dr. Morris, your pearls.
Maybe this mic is hot, so I can, I'll just stand here, and I think you have heard a lot about what I've said from other speakers. I think the people in the audience in Copenhagen who are academicians by now, I hope, have realized that doing grant-funded research, basic research knowledge generation, which is the reason universities exist, is different from a startup where the goal is creating a product which creates value for investors. And these are real people with their money, and they want to see that your product increases in value and provides them with a return on investment. Publications, nice to have. Return on investments, crucial. So there's a big cultural difference.
Thank you.
Before founding the startup, I really do firmly believe you need to know what the endpoint of your startup will be. If you don't know that, you don't know where you're going, you'll never get there. So you should really have a pretty good idea of what I'm talking about therapeutic, what your therapeutic will do. That would be the claim on the label. Phase 3 endpoint, unmet need indication. And I can't remember the other. We'll just shift it to somebody else. That's enough for me.
Your time, your pearls?
Oh, I don't have my pearls here. So I think I just echo what everyone has said, and I think we've all kind of repeated kind of the same themes you've heard is you've got to have a great team, you've got to have great medicine, and you've got to have passion and accept what you can't change. And so for me, why I do this is because I can't imagine, I've been practicing law for about 14 years. I can't imagine waking up every morning and working with a tech company and building an app that disappears in 4 minutes after you take like a photo. I love working with the great minds that are in this room and other boards that we worked with that are trying to make a meaningful difference in patient outcomes and being a very small part of that. I think it's very exciting. It is about making money, but also sitting in the board meetings with Dr. Miller and people, his counterparts. It's very inspiring. When you take these companies public or when you work with these companies' financings, the level at which the physicians interact is just it's what's kept me a corporate lawyer all these years. And there have been times, and I think one of our other speakers said, you leave corporate law, but it has been working with this amazing team that keeps me doing what I do, and I hope you all will start some companies someday and make some meaningful differences. If there's anything we can do to help along the way, we're happy to be part of the system.
Thank you. Stephen, do you want to go on? Oh, Dr. Harris. Are you still with us?
Yes, I'm still with you.
Okay, would you like to talk about your pearl, sir?
Sure, I was hoping that we'd be able to maybe engage and maybe get some questions from the audience, but if you just want some summarizing statements, I just want to say we at the FDA, we recognize that technological advancements have a huge potential to make a meaningful difference in lives and truly address major public health issues. And our programs and innovative thinking, innovative regulatory programs, smart regulation really is intended to help support innovation and ensure safety and effectiveness for the populations we serve in the US. Thank you very much.
Dr. Perez, yeah. Dr. Morris has one question that he wants to ask.
Hello, Dr. Perez, it's Dr. Morris. I'm not sure I understood you clearly with the breaking news that the FDA is now going to be called Food and Device rather than Food and Drug. So drug has been eliminated?
I was completely kidding. That was completely
okay.
Much better delivered in person. This is what happens when I'm translating over a continent. But the point there is that people forget many times when we have FDA, Food and Drug Administration, that we do regulate devices and all the technology is used in medicine every day. Over 190,000
the White House does strange things. So,
okay. Thank you. Stephen, do you want to go with your speaker?
So it's our turn. I think if we can get the slides up. Eddie, you'd be first.
Yeah, so the three takeaway messages from my perspective is that, if you have an idea, you should try and target a serious and life-threatening condition with unmet need, i.e. no effective or approved therapies. If by doing that, then you will not only get the attention of the regulators, but also potential investors. And this is what that was one of the factors, considerations when we decided to go to develop clazakizumab for the treatment of chronic active antibody mediated rejection. And then with that in mind, when we went to talk to the regulatory authorities, the FDA, CHMP, they were very receptive, and it enabled us come up with a clear development plan, not only to, and that was also very helpful in attracting corporate partners and potential. We did talk to a lot of VCs as well, but at the end of the day, we decided to go with a corporate partner. And then, you know, the regulatory authorities, because of the serious and life-threatening condition that we were targeting, they agreed not only to give us accelerated approval, you know, on the basis of a single pivotal trial. As you know, normally you would require 2 pivotal trials, and we could not only have to do, we could also enter straight into phase 3, not having to do phase 2. We leverage all the safety data, all the non-clinical data from the previous studies, and then the regulatory authorities basically allowed us to move straight into phase 3, which is where we are now. And then finally, as I said earlier, you need to form a team of, you need to collect professional, dedicated people to help you out. And that's what we have supporting us in Vitaris, although the 4 of us, although there's only 4 of us as permanent employees.
Thank you, Eddie. Then we'll pass on the word to John. John, your takeaway message, please.
There's an awful lot that I wrote up here, but what I would suggest focusing on really is probably the second bullet point, right? Which and I would summarize that by saying know the context in which your business operates. Obviously know your customer. That's a cliche, but know the context in which you operate. And so for us, providing donation and transplant software, the whole point of not negatively disrupting the work of transplant surgeons and donation, and instead collaborating with these partners to develop more efficient and more streamlined processes. Whereas in other industries, you absolutely do want to entirely disrupt what is currently being done. And so it's knowing the context within which your business operates. I mean, that's probably the best way of summing that up. The other stuff is a lot more detailed technically, and if anyone wants to start a software company, come talk to me afterward, and I'll be happy to share that with you.
Thank you, John. And with that, we'll pass the attention back to Stanford.
Dr. Miller, your pearls.
Thank you. I don't think I need to read my takeaways. They're pretty self-explanatory, but I can open it up for any questions. Maybe just one comment is, as exemplified by many of the speakers here today, the technology resources that we have now are just amazing. And the opportunities are great. And the ability to move things quickly from the lab to the clinic have never been greater. And I've seen, I know we have an FDA representative here. The FDA policies with regard to therapeutics in the past 10 years have really been phenomenally good, all moving in the right direction. Accelerated approval, breakthrough designation, orphan disease indications, et cetera, pediatrics was mentioned. These things have really, really primed the pump for innovation. When things happen at FDA, people pay attention, of course, and that's a magnet for more research and development. So I applaud what's been going on there. And really just the awesome amount of science is just spectacular.
Well, if there are any questions from Copenhagen or Stanford, please fire away. I gave my summaries, so I guess I'll just give 2 points. One is very much from the head and very sort of practical and scientific, and one is more from the heart. The first thing I'd mention, which I didn't mention before, is go to the FDA early. A lot of investors, especially around Silicon Valley, who are used to the tech culture, are quite frightened of government regulation. And obviously, you know, government can be slower than entrepreneurs like. But in my experience, to echo what Richard said, the FDA has been an amazing organization. You know, for a big organization with a lot of challenges, they've evolved quickly. And I found them very rational, very data-driven, really thoughtful, and really wanting to do good. So it's partly affected by the fact that we just got breakthrough status for Signatera. but when you go to the FDA early and they guide your clinical trial plans and have very bright, thoughtful people helping you get through the regulatory process, it's a much better thing for the company and a much better thing ultimately for patient care. So don't be afraid of the FDA, because they are different to what one typically understands of government agencies, certainly in my experience. And then the second thing I would say is kind of echoing the theme of my talk. You know, we can't explain concept. We spend so much time on science and amazing power of deep learning and cloud computing and all the things that we can do in genetics, looking at single cells, selecting embryos with all sorts of disease propensities, detecting diseases much earlier. There's an amazing growth of power. But we shouldn't let that get to our heads too much, because we cannot explain these most basic principles of the world. We don't know where time began. We don't know where space ends. And the answer, I think, that's emerging more in society is there's a growing spirituality. There's a growing sense that there is an answer to where space ends. It's a place of peace. It's a place of God or Dharma. And somehow, let your heart guide you as much as your head, because it really will make you more robust, and it's more likely to help you do good. That's it.
Thank you. I just want to conclude saying that, first of all, thank you so much to all the people who have worked so hard. Kelly, our Carla, Lisa, all the team, audiovisual, to put all this together, Gordon and his team. The most important, this meeting was about probably not necessarily to bring new generations of thoughts, but just to say that everybody of us here in this room has a function. I wanted to invite specifically coordinators and nurse practitioners for one reason. The questions start with you. What are the gaps that we have? Medical records, patient care, what are the gaps? And we have people here that have technology that can come and say, hey, let's talk about it. And let's start the next generation of care for the patients. I have the privilege to have Dr. Osorio and the team from Sutter and CPMC. Why are we doing this? Why are we invited? We cannot be isolated anymore. We should be able to all, not just regionally, state, internationally, get together and find solutions for our patients, because we have common problems. We have the trainees here. Why the trainees are important? You are the ones that are going to be generating the next generation of answers for us. Unfortunately, we have an institution like Stanford, we have an institution like European Society of Organ Transplantation that provide this forum to create this platform, not just for now, but for the future, to continue talking, having dialogues, and most important, find solutions and be the improve the healthcare that we have for our patients. Thank you from Stanford.
Thank you, Waldo. Thanks to all the speakers in Stanford, but also here. It was a privilege to put this together. Thanks again to Randy, to Annalisa Ponke. I think it was a pilot that was unique, and I can only hope that we can build on this, we can develop this further, and come back with the next edition. I think these stories were extremely inspiring. Still people here, despite the late hour, are following. There's many more through the ESOT Live channel, and I'm sure there's even more that will look at the recordings of this session. So thank you all for doing this. It was very inspiring. Certainly, I have the impression it was huge, new, something to build on. Thank you, Stanford.
Thank
you guys have a great day. I hope you enjoyed it as much as we did.
Thank you so much. I'll take a picture of all of you. Thank you, everybody, for being here. Please have more food, and thank you for making the trip. Bye.
Hello and welcome to ESOT's Transplant Live platform. If you are a transplantation professional anywhere in the world looking for high quality education, you've come to the right place. Because ESOT aims to inspire, connect and innovate the global transplantation community. We do so by creating the most attractive educational content. Through face-to-face meetings like the ESOT 2019 Congress right here in Copenhagen, in print, and through this online platform that you have already discovered. And right now you are participating in something truly unique in the transplantation community, because we will be live for 24 hours during the 4 days of the ESOT 2019 Congress in Copenhagen. And what can you expect? Well, obviously, we take you live into the halls where the sessions are taking place, giving you the latest insights in the field of transplantation. We will also show you recordings of other sessions that are going on in parallel. We'll show you interviews and reports with some of the key thought leaders and opinionators that walk around in this Congress. And in between, we'll show you some great examples of the education that you can find in the Transplant Live platform and some of the best sessions of the Congress in Barcelona 2 years ago. So don't be selfish and share the URL esottransplantlive.org with all your friends and colleagues and tell them why you decided to sign up. And then just keep your browser open. You start once, stay tuned, because it's happening right now.